All right, we're up and running again. Sorry about that. Okay. So what these researchers like Hubel and Wiesel found was that the receptive fields of ganglion cells in mammals, so humans, cats, monkeys, and so on, have this centre surround organisation. And so what that means is when light falls on the centre of that particular cell, it's stimulated. If it falls on the surrounding area, then it's not stimulated. And so what the name of this is it's called spatial antagonism or lateral inhibition. So there's a diagram in the next slide which shows that a little bit better. So what you have here, the cell, that's the ganglion cell there, the whole receptive field. The outer part has got a little minus. That means that it's not responsive to a spot of light, whereas the centre is an on centre. And there's little pluses there to indicate that that's where the response is elicited. So what happens is this is the little light shown here by that little yellow circle is shown on the centre there. The ganglion cell is stimulated. Now, remember, you're not conceptualize this because it's all happening deep in the retina. You're not really, when you're shining a light, you're not shining it directly onto the ganglion cell because that's deep inside the retina. The connection happens via the rods and cones and then down that particular pathway, and eventually that ganglion cell is stimulated, if you know what I mean. So happens to fall here, it's hit the right cone, and it's all the synaptic connections have joined up, and eventually the cell is stimulated. The light falls on the center, and here is the diagram of the excitation of that cell. So it goes crazy, buzzing, very happy that a light has fallen on the center part of the cell. Now, if a light happens to fall on the outer part, which is the off surround, there's no stimulation of the cell. So nothing happens. And when nothing happens, it just means that there's no visual image transmitted. There's no electrical signal transmitted. To land precisely where the cell wants it to land in order for a visual stimulus to be passed down by the optic nerve. Why does this happen? I don't know. That's just the way that we're made. These particular cells are on center, off surround. Where there's a plus, it's happy, the cell gets stimulated, sends an electrical response. If it falls outside of that, no response at all. The response to have vision, obviously. But there's millions and millions of these cells all doing different things all at the one time. More reading on page 273 and 276 in Schwartz if you want a more detailed explanation, but you don't really have to get too hung up about it because you can get quite involved in this and get yourselves very, very confused. We said yesterday that the photoreceptors contain this photosensitive pigment which absorbs units of light, photons, or one unit of light, and that will then convert that into an electrical activity. And I explained this, that it is the first step in vision, and then eventually that all gets processed down the visual pathway. So what you need to remember is that the photoreceptors are slightly depolarized compared to a typical neuron, and polarization that occurs relates to how intense a particular stimulus is going to be. And this is called a graded potential. Now, I presume that you guys have done action potentials and this kind of stuff in other subjects. What subject does this appear? HBA. Oh, so that's in first year, isn't it? So you might have to dig out some of those notes to help remind you if you've forgotten some of these concepts. Now, this is what happens when a rod is hyperpolarized. That's the next set of slides. And I do need you to remember roughly the process that happens. Complicated and boring, but essentially this is how it all works. So what happens is we're in the dark. Rods prefer to work in the dark, correct? Yep. So sodium ions will flow into the rod outer segment. Oh, I've done it again. Through channels of the rod, and we know that ions flow in and out of cells through these channels, and that will cause depolarization. Now, when light is absorbed by a rhodopsin molecule, remember I said to you that rhodopsin was one of the molecules, was the molecule in rod that absorbs a unit of light. We talked about this yesterday, and then it doesn't send a signal about what the wavelength is. It doesn't care because we don't need to see colour in the dark. So when light is absorbed by rhodopsin, it causes blockage of the sodium channels and you get hyperpolarization. Now, the next step is that it relates to actually, I'm going to just go back and explain the whole situation from the very beginning, and we'll start at where all these chemicals change. This is where I find it confusing, and I know it's going to be a bit tricky for you, but hopefully you'll follow me through it. So, rhodopsin contains opsin and chromophore, and it's the opsin chemical that will determine how that particular rod will absorb light. The chromophore inside the molecule, inside the rod, contains retinal. Now, it's a form of vitamin A. So you know how they say you should eat carrots because it's good for your eyes. We're drilling right down into the rod photoreceptor, where this form of vitamin A in the form of retinal is contained, and you need your vitamin A for the purposes of all this electrical current and electrical signal. When retinal inside the rod is unbleached, it's in a molecular form called 11-cis up there. So this is an unbleached state. So this means that nothing's happening in the rod. That chemical is, or the process is 11-cis retinal. It's asleep. Then it starts to change its chemical composition once light hits it. So in this slide here, we have a dormant rod photoreceptor and molecule in its 11-cis state. So going to happen is a photon of light. Here it is, a little thunderbolt of light strikes the rod, and all of a sudden a whole series of changes are going to take place, and I'll tell you what they are. So now all of a sudden the rod will absorb that photon, or the molecule of rhodopsin will absorb that photon of light, and this 11-cis retinal is no longer called 11-cis retinal. It changes its chemical composition into something called an all-trans isomer, which is a type of protein here. All-trans isomer or all-trans retinal. And this then creates a series of chemical activations. It then activates transducin, which is a protein, and then that activates phosphodiesterase, which is another type of protein. So what we have here is nothing was happening. It's called 11-cis retinal. It's in the dark when it's asleep. Photon of light comes along, strikes it, and this series of chemical changes occurs. The 11-cis retinal chemical changes to 11-trans retinal. This activates these two types of proteins, transducin and phosphodiesterase. That's what's happening inside the rod, but many times over. Because remember, we've got many types of these molecules floating around inside the rod photoreceptor, not just one. So we're not talking about one rod, we're talking about thousands, millions of these little chemical changes happening all the time. And so this is that metabolic activity, which, as I said yesterday, is what needs, the debris of all this needs to be cleared away, and it's the what that gets rid of all the gunk, all the junk at the rods. The microvilli, which are part of the RPE, the little brushes. Yeah. Okay, so that's where we're at the moment. Now, the next step, once these two proteins are activated, the transducin and the phosphodiesterase, causes more chemical changes, and there's more molecule breakdown into something called GMP or cGMP. And then the outer of the rods will polarize. So that means now all of a sudden hyperpolarization means it's being stimulated. So all that stuff just to get a photon of light in there and get it working. This is quite complicated, right? So I guess for your purposes, I will summarize what I'd like you to know, because often this stuff does appear on the test papers, final exam. So